So friends, this program is coming to an end. Amar, we will say something in before we start.
Yes, like I would give like a small outline of the workshop.
Yes, please go ahead. Go ahead.
So, good morning, students. Good morning, professor. As we are coming to the end of the workshop, 21 days workshop, it was a pleasure to interact with all of you. Our BIIS-10 program announcement was published on 20th December 2021, as you all know. We received application of 238 for this program, out of which we selected 70 eligible students, and 15 were from the aspirational district, 43 students from Tier 2 cities, and 12 students from Tier 1 cities got selected for this workshop. Due to the third corona wave, there was a little delay in starting, and we started this workshop at 15th February 2022. A total of 47 sessions of invited guest lectures from various disciplines were conducted under different themes like grassroots innovation, life science innovation, entrepreneurship, startup ecosystems, IPR and drug regulations, biostatistics, microbial biotechnology, agriculture biotechnology, veterinary sciences, phytochemistry and pharmacognosy, medical devices and point-of-care diagnostics, bioinstrumentations, bioanalytical techniques, stem cells, bioinformatics, in silico drug discovery, and machine learning. We had our guest lectures from several top institutes of the India, like IIT Madras, IIT Bombay, Indian Institute of Science, Bangalore, ICT Mumbai, NIPER Ahmedabad and NIPER Hyderabad, National Institute of Plant Genomic Research, Gujarat Biotechnology Research Center, Central Leather Research Institute, Chennai, industrial specialists from Anchrome and Biorad, National Cell Center, Science Pune, National Institute of Animal Health, Hyderabad, Center for DNA Fingerprinting and Diagnostics, Hyderabad, Institute of Himalayan Bioresource Technology, Palampur, Manonmaniam Sundarnath University, GB Pant University, like this. At the end of each week, we were having assignments.
And we also had people from National Innovation Foundation.
Yes, sir. At the end of each week, students were given assignments and quizzes based on the lectures we were conducted. Students were introduced to the grassroots practices documented by Srishti and Honeybee Network, and were guided and asked to develop project proposals. This is the outline of the 21 days workshop we have done so far. And I am handing over the session to Professor.
Good morning, friends, once again. Some of you missed yesterday evening session. I do not know why, but we would have wished if more of you had attended the session last evening, on the presentation of synopsis, in which we gave the feedback to those who presented, and some of you were advised to revise your proposals. Have some revised proposal been received, Amar?
Yes, Professor.
How many?
3 to 4 proposals, revised proposals came. Professor.
Because everybody was asked to revise, if only 3 to 4 have been received.
Yes.
This is very distressing, because we will not be able to ask anyone who has not revised to present today, no matter how useful the idea was. And this prevents you from sharing your work with everybody else in the valedictory function, which is being webcast all over the country, as you know. So your work is being noticed by the biotech scientists, entrepreneurs, policymakers, and if such opportunities are missed, then surely something is lost. So my advice to you all is that let us not miss or undervalue such opportunities, because these opportunities provide us a unique occasion to make wider awareness about our work. So it's important that we share, make whatever modification we can, and improve. The purpose of this is to build a capacity of our undergraduate students. With exception, we also accept some postgraduate, and rarely we have also accepted some school students. We have made exceptions on both the sides. And please spread the word around that while this is intended for students of undergraduate course in life sciences, medical devices, bioengineering, biotechnology, agriculture, pharmacology, veterinary science, various other disciplines of life sciences or linked to life sciences, but we do accept exceptional cases, the students of postgraduate and school, if they show a brilliant potential. Having said that, let me also say what we were trying to do. We were bringing scholars of repute, and I understand that some of you must have found it difficult to cope with the complexity of the lectures that they gave. All these lectures are available on YouTube channel of Honeybee. Therefore, even if you did not follow everything, someday in future, when you need to, you might go back to them. When a subject is brought back in the class in your college, you can always refer to that. And opportunity to bring so many outstanding scholars from all over the country at one place probably may not be accessible to any one of you in your college, because perhaps your college may not be able to mobilize all of them. But we, with our social capital of Honeybee Network over the last 35 years, have built a huge respect among the professional community because of the work that Srishti and Honeybee Network and other institutions of Honeybee Network have done, GYAN and NIF and all of them. So therefore, we are able to request and persuade colleagues from IITs. You mentioned, I think, IIT Bombay, IIT Madras, IIT, many other IISs or NIPAS institutions. And this resource will remain available to you forever. So please look at the lectures even of the previous BIIS, previous 9 BIIS and the 10th BIIS. All of these are available to you. All these lectures, Amar, are uploaded, isn't it?
Yes, Professor.
Yeah. So what we could do perhaps is to make an index of all the lectures with their links.
Okay.
And send to all the alumni.
Sure, Professor.
Now, my dear friends, you will become alumni of BIIS and Honeybee Network from today onwards. From tomorrow, you will be called as alumni. Though those of you who would like to work on a proposal, and of course, all of you have to submit a project report to complete the requirement of reports protocol. But 10 of you, as I mentioned yesterday, I'll repeat, 10 of you would get 1 lakh grant if your maximum 10, it could be less, whose protocol or whose ideas are considered outstanding. But the invitation to come to our lab and work is extended to all of you. So there's no restriction. For those who get fellowship, they can use the fellowship fund for sustaining themselves and for meeting the cost of research and all that. For remaining, if we find that your proposal is really meaningful, we'll provide you local hospitality, but other costs you have to meet. We will not charge you for the chemicals and other things. That lab will be here. But you will have the opportunity to come here and work on your project if you are convinced that you genuinely desire to validate and or value add in the knowledge that community will benefit from. So this invitation is extended to all of you. I would also suggest that by now you know what Honeybee Network does. Will anybody summarize in 1 line what Honeybee Network does? Anyone?
So it stimulates us to innovate, and it helps us to connect to our grassroots to help our people.
Anybody else? Little more than that?
And it helps to bring back household practices and grassroots practices to a higher level.
Higher level for what? To do what?
To do more sustainable technologies.
Okay. In which domains are we more active?
Agriculture, health sector,
Microbiology, veterinary, and pharmaceuticals.
Okay, this is technological. What else? Which are the other verticals in which we work? Mainly, broadly. Obviously, we work in education, isn't it? You're all students. And we are bringing you together. When we are bringing you together, we are providing award and fellowship to the PhD students. Sitare Gyati Award and Sashti Gyati Award and function is going to take place either this month or early next month. And so that's another thing that we do. So we also work with the school children, we work with teachers. We look for innovations in education. We look for innovations in technology, both from grassroots, farmers, artisans, mechanics, and for grassroots. Third, we look at institutions, formal and informal. So when we created the note for BIIS, we contributed to creation of the BIIS when I was at IMA along with my colleagues. So we also help in institutional building in the formal sector, but we also work at common property resource institutions at the community level, urban as well as rural. And fourth is technological, institutional, educational, and cultural. We also work with the knowledge system, cultural knowledge, the knowledge of the food. So we've organized Satvik, as Chetan must have mentioned to you and Ramesh must have mentioned. We also have an incubator. And this incubator, Sanctuary of Innovation and Incubation and Entrepreneurship, is set up to help students like you, but also others, farmers, artisans, mechanics, to take their ideas through value addition to the market. Social market, that means as a DIY solution, do-it-yourself solution, or commercial market, that is as a priced product. We organize Shodhyatras. We have walked in every part of the country. So that's another thing that we do. We also create databases, as some of you have already seen. We create databases. So we have created many databases. You must have seen at honeybee.org, we have a medicinal plant database for more than 200 plants, and their uses for agriculture, veterinary, and human are documented. Of course, it's slightly old now, so literature may not be up to date, but the main characteristics will remain there. The chemistry of the plant will not change, so sufficient information may come up. So we create databases, open databases. Anybody can use them. Anybody can search them. No permission is required. We have created techpedia.in. Anybody knows about what techpedia.in has? Anybody?
So patents.
No, techpedia.in has, anybody else knows what is techpedia.in? Techpedia.in has more than 200,000 title or abstracts of student projects from more than 600 institutions pursued by more than 500,000, that means 5.5 lakh students. And why did we do that? So that no student does what has been done before. So we have techpedia.in. Then we have a techpedia.in/patent, which has, or gyan.org/patent.php. Both are same databases. And this database has helped more than a million abundant patents from US. Abundant meaning these patents were granted and then fees was not paid at 3.5 or 6.5 or 11.5 years. So they were abandoned. Now they are open access. All these databases we have created so that you can access them and improve the quality of your research, quality of your products, quality of your services. Why is Government of India, BIIS, DBT investing so much resources in building your capacity? Why would they do that? Anybody? Of course, one is to build your capacity, but what the long-term purpose? Anybody? The purpose is to create a pipeline of entrepreneurs. We hope that some of you, either now or during your post-graduation, will translate your research into enterprise. And then you will apply, as Shilpi had explained to you and Manish had mentioned, for big grant. Big grant is for 50 lakh rupees, even sometime on the concept only. Even if you don't have a proof of concept, even then you can apply for it. If your idea is found rigorous, if you can defend it before a jury, you might get that. Or Gyati Award, which is for 15 lakhs for 2 years to 15 students. You can apply for Gyati Award as you move along. You can apply for big grant and many other opportunities that are available. So why are we doing that? We are creating a pipeline of bright, brilliant, socially committed and responsible students who combine excellence with relevance. Excellence with relevance. So you should be outstanding, but you should also be socially more useful. Your contribution to the society through your research, translational research that doesn't remain at the conceptual level or theoretical level, but is translated into practice. That is called translational research. That research which is operationalized for application at societal level can generate two kinds of outcome. As I said earlier, I'll repeat, DIY solutions. So some of the practices that you're going to work on, once you validate them, you elaborate them, you can build a protocol, we can share, we will also share, anybody can access them, anybody can replicate them, save their cost, improve their knowledge, improve their effectiveness. And some can go as market-based solution. That means you can set up companies, you can set up startups, and of course, as you know, you must have been told, there are so many incubators, C-Camp is available, and you can get benefit of the those platforms for taking your ideas forward. So this is the purpose why we are organizing this. So please understand that all the energy that Srishti has put in, it has put in to bring the best out of you. Best out of you intellectually, best out of you morally, and best out of you socially and emotionally. Because we believe that once you get committed to the values of Honeybee Network, and what are the values of Honeybee Network? One, that we don't make, whenever we learn something from someone, we acknowledge them. Second, whatever we do with that knowledge, we share with them in local language, if possible, personally, but otherwise we send it to them. Third, we cross-pollinate the knowledge. So let us say, Prithvi will find something useful that Saurabh has done. Saurabh will find something useful that Abhishek has done. So you will share with your respective social network, with communities in your region, knowledge that you find useful, even if produced by somebody else. You will work like a honeybee. You will do the cross-pollination. And fourth, if you develop any value-added product and you get some monetary compensation or consultancy or honorarium or royalty, you will share a reasonable part, fair and just share of that income with the people from whose knowledge helped you to set up your enterprise of your product. These are the key principles of Honeybee Network. We also believe in open innovation. So as I mentioned earlier, I will repeat that we expect all of you to monitor upload to download ratio. Do you remember what is upload to download ratio? Anybody remembers? Anybody remembers? What is upload to download ratio? Unmute yourself and tell me. Nobody knows? Amulya? Mohan? Tarumai, what is download to upload ratio or upload to download ratio? What is the meaning? What is the implication of this? I must have mentioned, I'm sure.
So, is it like whatever we learn, we give back in some way?
Well, if you put it in public domain, either through a blog or otherwise, then that is upload, correct? And the download is whatever download you do for papers, videos, and other things. So the reality of our life is that we download a lot. We upload very little, isn't it, Arushi?
Yes, sir.
Yes, sir.
So we would like to change this. And if this has motivated you, then please share your learning. Tell other people. Every one of you should write at least one blog. And we can upload it at Creativity at Grassroots blog or our own blog, so that your feedback, your learning, your critical suggestions about improvement, and some specific insight that you learn, not just general, oh, I found it very useful. That doesn't help. But what did you learn? And how did it shape your ideas, your thought, your future research direction? If some sessions in the BIIS made you more curious than what you were already, then that is a great thing. We were very happy with that. We would like to know, and those faculty members would like to know that their lecture here triggered a curiosity in the mind of some of you, such that you may have decided that, okay, I'm going to do my post-graduation in that branch of biotechnology. Wonderful. Isn't that nice? It's nice for the society, nice for you, nice for us, and nice for the concerned faculty member whose ideas triggered your imagination, or at least reinforced your idea. Maybe you already were thinking about that, but the BIIS has reinforced it. So I'm very happy that all of you now understand a little better about what Honeybee Network does, how Srishti tries to provide support in different verticals. And there are a lot of databases which all of you can use. Database on medicinal plant, database on common properties, database on farmers' innovations, database on practices. In fact, some of you those who have not pursued should know that NIF, National Innovation Foundation, has shared 135,000 practices in open source at their website. 135,000. Pulled from various Honeybee Network volunteers. So that's a kind of a national register of traditional knowledge and some innovations. You can access that. All of you should consider, you should find time to look at that database also, what NIF has developed. So please understand that all of this, the open access databases on abandoned patent, on student projects, on medicinal plant, on common property resource institutions, CPRI, on open access database of NIF, database of Polytechnic project at Gyan Nidhi, all of these are open access which you can access. Now we come to the last part of this part. We are going to get Dr. Shekhar Mande for a brief interaction with you, because he is in the field, and depending upon the network, he has promised that he will call us at around 11 or 11:15. He's in Palampur, Himachal Pradesh. And if he doesn't get the Zoom connection, he will call me up, and then we will put it on the speaker mode, and all of you will be able to listen to him. He is the Director General of CSIR. But he would come later. So, Amar, are we going to have some presentations for the final one? Those who have revised?
Yes, Professor.
So why don't we have that now?
Yeah, we can start, Professor.
But before we start the presentation, anybody has any comment, question? We also want your feedback today. So we will have feedback maybe after the presentation by maybe after 11:30 or so. But if any one of you has any quick comment on whatever I have said, please go ahead. Or comment, question, criticism. Yes.
Sir, I have a question. Like now we have constructed a proposal or a protocol for a project. So if we need a mentor like to construct the final protocol or design it, because we don't know the last step of it. So can we get, sir?
Yes, you will get all the help from Sashi team, and in areas where they think they need to contact somebody else, some other faculty members, either from NIPER Ahmedabad or somewhere else, I will facilitate that. Any
Sure,
one
sir.
of you or each one of you who needs guidance to take your research forward will get that. We are committed to provide you that.
Sure, sir. Thank you.
Yes, yes. Feel free to write to our team, and whenever you feel you have not received response within, let us say, 2 to 3 days, write to me. Okay? I promise and commit to you that every mail of yours will be replied. Next,
Okay, sir.
please. Few days back, one student had written, Amar, was she from the participant of this program who had gone to a village?
Yes, Professor.
Who was it?
I only, sir.
Oh, you were there. All right. So, Amulya, I would like to mention that your mail, I read it with great interest. Have you further developed your thought what you want to do with that community?
Yes, sir. I have like it's my plan for future because I have to contribute to that society somehow. So I'm working on it. After finishing my studies, at least I have to go and research there and do some contribution to that society, sir.
And we will encourage you and support you for your documentation purposes and also for your on-farm research purposes.
Thank you, sir.
So during the summer, when you have time and you're planning to go there,
Yes,
be
sir.
in touch with us. We
Okay.
will also give you a format. Amar, please circulate the format that we have made for Village Knowledge Register.
Okay, Professor.
You can circulate that. I recently made, that is about a few weeks ago. And if you don't have, write to me, I'll send it to you today. So that format will be shared with you. Those of you who want to go and spend some time with communities and create a village knowledge register for your own native village or any other village, you're welcome to do that and submit a copy to us so that we will also be able to share with the larger world around the world. So that community and its knowledge system, with whatever restrictions they want to put on that knowledge, with their prior informed consent, of course, will be uploaded. If they say that, look, this part you can share, but this part you don't share, then we will not share that part. So prior informed consent is very fundamental for any knowledge which we collect from the people and we share it with the larger world at large. Okay. So any other comment or question? Any other? Some of you did not present yesterday evening. What was the reason?
So I had a prior commitment, like it was decided 4 or 5 days ago that I was supposed to go somewhere. That is why I could not come.
So you could have sent a message, no?
So I did send a message.
You did it? Okay. Who else? Anybody else? Who abstained without prior information? Would you like to help us understand what happened? Because for us, if somebody doesn't attend a session, it's kind of a feedback to us that maybe you're not finding it interesting enough. In that case, you should tell it openly. And we should let everybody in the country hear that there were some participants who could not find enough excitement in it. Of course, that is a social audit. We are all accountable, isn't it? So please don't hesitate in saying that if you think that was the reason. Did anybody not attend because they were not finding it useful? Raise your hand or unmute yourself and tell me. Be honest and frank. Professional integrity demands that we say what we believe in. And it is all right if you didn't find it useful. It will help us to improve it. The next BIIS is starting on Monday. And Dr. Renu Sarup, former Secretary DBT, who had also addressed you, will be giving a talk at 7 o'clock tomorrow evening, and Monday evening. So all of you who wish to attend her session, once again, because she will bring some new insights, you're all welcome. We will send you the link. And we'll send you the program for the next BIIS also, so that just in case you want to attend some session, Amar, allow them.
Okay, brother.
Yes.
In case they missed, they can attend.
Yes, exactly. So if you have missed some session and you want to attend them now, you are welcome to do that because of your exams or other reason. All right. So if there is any other comment, Murli, Tarumai, Kenza was there, Rohan, Anjali, anybody, Srishti, anybody wants to make any comment on what I have said so far, or any question that is there in your mind about how would we help you to take your ideas further? You're welcome to speak now.
Good morning, sir.
Yes.
I had presented my proposal, the synopsis yesterday, and you had suggested some changes. So, like according to what you said, I looked for additional sources, but then I did not come up with anything constructive to build upon it. So I was just thinking if I should change my proposal idea.
What was your proposal on? Please remind me.
I think it was on like formulating an anti-weed herbal one for like coriander crop and for like other crops for the same soil.
My suggestion was, and I have told you, and I will show it to you just now. I told you to look at the database. Did you look at the database?
Yes, sir. Yes, sir. I did look at the database.
Couldn't find anything on weed?
Weeds I could find, but then for coriander, I could find, but then I'm not very I didn't know what I could use from there. I was out of ideas. You could say that.
No. What I'm saying is, let me put it here. Weed. All right, let me show it to you. So I'm sharing with you the screen, which I told you yesterday to access at honeybee.org. Didn't I tell you that?
Yes, sir. I checked this only.
So if you look at it, you will find that there are many practices. One is crop rotation. So if you grow, let us say, lucerne or bursine, because they're cut many times, they won't let the weed set seed, and therefore they will not be there in the next cycle. We have many other practices. Let's see, what is this? Now this is the use of a weed. Let us go further. So coriander controls weed. Look at that. So striga weed in sorghum is used called coriander seeds are planted between the rows of jawar, and once the coriander plants are big enough, the strand twists themselves around striga weed and suppress its growth. This way the weeds are finally eliminated. So interesting, because they don't affect the sorghum now. Otherwise, striga is a creeper. It winds around the plant and does not let it yield much. So if it grows around coriander, it apparently gets suppressed afterwards and does not harm jowar crop. 1997. Interesting idea, isn't it? So you can see there are many other practices. Let's look at this practice from cumin controls weed. So Cynodon dactylon is a perennial weed, and there's a farmer, Dev Shibhai. What he believes is that if you grow the cumin stem spread in the field during monsoon, then it results in the stalks are allowed to decompose over a year, and later on crop can be grown without any problem of Cynodon. So if you can afford to do that, why not? Not immediately after. cotton shells for weed control. Look at that. So there is a practice here where cotton shell are applied to the field and they help in weed control. So there are many practices. You can look at them, select some of these or others, doesn't matter, and develop your proposal again. Is that all right?
Okay, sir.
So you can still try to do what you want to do. One treatment can be what you have already thought about. Even if it proves wrong, doesn't matter. You will learn it empirically. But you should have few more treatments so that your effort becomes more meaningful. Any other comment or question, please?
Sir, you had asked me to revise my proposal yesterday. My project was about chromium uptake using the components of Triphala, that is using Harad and Amla. So to bring novelty to my project, since the time was less, I could not do a wide scale review of all the articles present. So what I did was I found out the phytochemicals present in Harad and Amla, which is responsible for chromium uptake. That is hydrolyzable tannins like elagitannin and gallotanic acid. Yes. So I then, as I learned from BIIS to use bioinformatics tools and databases, I found out a database called IMPPAT, which is Indian Medicinal Plants Phytochemistry and Therapeutics Database. And using that database, I found out which all plants have been studied to contain these hydrolyzable tannins like gallotanic acid and elagitannin. And accordingly, I have found out a list of plants, which Indian medicinal plants, which contain these hydrolyzable tannins. And then then after finding one plant, I'm looking up whether there has been studies on chromium uptake on that particular plant. And because I'm certain that it contains hydrolyzable tannins, I'm sure that it will work, and we can study the details of how it takes up chromium. And I have also thought of some experiments which I can do. Like there has been a lot of study on agricultural plants facing stress due to chromium presence in the soil. So I would like to do an experiment using rice plants, a control experiment in which rice grows in chromium-rich soil, and other potted plants in which
No, this is very good. I have put it down on the chat one more database. Have you heard about that? This is a database called as IMPPAT.
Yes, sir. That is the one I'm
That's the one you're referring to?
Yes, sir.
Then there is one more I will like to tell you. And this is the database that FRLST has developed. And this is the www.medicinalplant.in. So let us see if this is accessible openly. And if so, you can all search it. This is the one. So I put it here. All of you should take down these two because they will be useful to everybody, not just Ayush. So this is medicinalplants.in and PubMed. This other one I will also share. This one. And this is a good idea, Ayush. What you did was the right thing to do, to look at the compound and then find out the plant which have those compounds. And if they are easily accessible, go ahead. I mean, try those plants as well.
Yes,
isn't
sir.
it? So that's a very good.
Sir, yesterday I was looking at seeds. Then I realized that if I want to do it on a large scale, then seeds, the biomass of seeds is comparatively less. So I have started looking at the barks of trees. Like there has been no studies on chromium uptake using Madhuka indica bark trees, which are also rich in tannins. So also looking at.
In fact, all barks are rich in tannin.
Yes, exactly.
All barks, not just Madhuka indica, all barks.
Yeah.
Now, there are some barks which have more tannin and are used, let's say, for processing leather.
Yes, sir.
Which are a little more acidic also in nature, so you have to be careful. But Nevertheless, tannin by itself would not be solving your problem. You have to look for which kind of tannin. Yes, and which concentration. And if you are doing it in water, then it should be human safe.
Yes, sir.
Not all tannins are human safe. Right? So
Yes,
you should
sir.
be very careful about that. But this overall, I think the right direction in which you're moving, your mind is moving, is very correct. And I would encourage all of you to take a cue from here and look at these databases, the one that Ayush mentioned, and I have added one more. All of you can access these databases. Our team should also add these databases to the list of resources that are available for BIIS. All right?
Okay, boss.
So please make a database list on BIIS site.
Okay.
So that anybody and everybody, all the students will be knowing about what are the open access databases in the world. There was also a NAPRA alert, which I used to access earlier, Natural Product Alert Database. University of Illinois, Chicago has developed. Professor Farnsworth, a dear friend of mine, had developed it. It may not be free anymore, but still check it up and make it. So you can mention in two parts, paid databases and free databases.
Okay.
Right? So please make a directory of all databases. And I would encourage all of you, the students, to make list of open sources. There's another site, Intech, which has a lot of videos, free videos and chapters and book chapters, all open access. Amar, did you share that with them this time?
No, not at Professor.
No, because I had shared with you and I have mentioned that we should all tell our students they can get all full books actually for free access. So please, I'm going to share this with you now. I'm sorry that we did not share with you earlier. intechopen.com. This is this website, and I would encourage you, all of you, to use this. Make it a permanent resource for all of you. Please make it a permanent resource.
Okay.
Sorry. Come up. Let me copy again. And all of you should use this. All of you should use this. Because nowhere else you get books, downloaded books, intechopen.com, intech, open.com. This is a site where you get free books. Open science, open mind. That is the slogan of this site. And let me share it with you. This is the world's leading publisher, which puts all the books in open access. Now, every student of biotechnology. And please share it, Amar, with all our alumni.
Okay, Professor.
Because these are resources which will give them books on nanotechnology, on geodisk science, on renewable energy, on such, whole range of subjects. You can get full books, open access books, all of them. 5,700 full books, which have been cited by more than 100,000 people. And of course, millions of visitors come every month to these books. So the advantage of publishing here is that your work gets cited a lot. Open, fast, peer-reviewed book series, you can publish them also here. So please look at, and they got Nobel Prize winners who have published books at Intech Open. So you may not get royalty from here, but you get visibility, and you get feedback, and you get followers, and you get new friends. Understand? That is what this does. So please use this resource. Use this resource. It's all yours. It is all yours. Life sciences, health sciences, social sciences, physical sciences, engineering, and technology. All these books are accessible.
Professor, recently IIT Kharagpur has developed a National Digital Library of India.
Correct.
It has more than 60 lakh books. So I am sharing the link also here.
Yes. And then you should also share the link of NPTEL.
Okay.
There are open courses, full-fledged courses, which you can actually take and add to your CV and add to your knowledge. And these courses are taught by IIT and ISC professors. So NPTEL is another source, IIT Kharagpur source I'm aware of. You can use that also. And all of this, nptel.ac.in. It's the MHRD initiative. And you can come across whole range of courses here. All right. Courses by IITs and ISC. 1.6 crore. Let me show you. It's a very powerful source. And I'm happy that we have been able to recall these today, because it would have been sad if I hadn't shared this with you. So 1.3 billion views, 37 lakh subscribers, 2,300 unique courses for self-study. You can take these courses, complete the assignments, and once you complete it, and these are evaluated by the concerned faculty members, they have periodicity, of course, when they offer these courses. And you can take these courses from any one of you. And these courses are by faculty from IITs and ISC. And there are 15 lakh people out of 1.6 crore, almost 10%, who took exams also. 3,500 MOOCs completed. Industry Association. So all of that is available to you. Please don't miss it. Swayam platform, National Program on Technology Enhanced Learning, NPTEL. Are you able to see all of it? And this portal has had more than 471 million views and so on and so forth. Please use this. Enrollments are open for January to April semester. Of course, this is old now, but you can do that for next semester. And you can take courses along with the students, other students, and build your own knowledge base. So NPTEL, IIT HP NDL, National Digital Library, all of these are open access resources. Why are we sharing this? Because we want you to learn. Because the course will end today. BIIS will end today. Amar, in the final letter that you will send to all of them, please add these sources once again.
Okay.
So that they get all of them in one link. All right.
Okay, Professor.
So let's have, is there anybody else, any comment before we proceed for presentation?
Sir, can I tell
Yes,
my
presentation.
idea?
No, we will have presentations one
Okay,
by
sir.
one, but only those who have revised. Is that clear? Those who did not attend yesterday and have not, of course, revised also, did not present, then they will not get a chance today. You can submit your ideas, of course. We will review them and send you feedback. For presentation today, it is a requirement, of course, that you must make a presentation a day before, revise them, and then submit. Because that is a protocol, that is a minimum protocol we should follow for a responsible study program. So Amar, please invite those who have revised.
Yes. Tarmoy?
Yes, sir.
Yeah.
So, shall I share my screen?
Yes.
Okay, sir. So the sharing is disabled.
Yeah, wait a minute. Yeah. Now try now.
So my screen is visible?
Yes.
Okay, so my project proposal is about finding a potential plant growth promoter and a biopesticide at the same time, which is derived from the citrus species. And these ideas are adapted from the Indian traditional practices. So my problem statement is, of course, developing a new and potent herbal solution from various parts of the citrus plant, like the leaves, flowers, and the fruit peel. So reason behind choosing my topic is, as we know, like today, due to the increased demand of the food and a lot of innovation by the pests, so farmers use various chemical fertilizers and those chemical pesticides, but they are harmful, we know that. So, but there are various farmers who uses different traditional practices which are completely organic. Like from the honeybee network itself, I found that Sri Lalaji Mafaji Vagela, who uses the lemon leaves and soak them in the water and sprays it in the gourd, which helps in the fruit size increment, and it also takes the fruit from out of the pastes. He also uses the flowers and the peels of the lemons as a seed germinator for cabbage also. Another knowledge shared by Sri Tadvi Jetavai Veerabhai, he uses the citrus flowers as an insecticide against Helicobacter armigera, which is the cotton bollworm, in his cultivation of pigeon pea. And he uses it for many years, and he also gets fruitful result from it. So knowing this potential of citrus, I think that there is a potential to develop a product which can act both as a plant growth promoter and also as an insecticide. So from this, herbal solution development will be my objective. And for this, my methodology will be as already shared by the farmers. Their methodologies are already mentioned here, and I think that there is a possibility that we can mix the compounds of citrus leaves and flowers together and see how it together can affect the growth promoting and also killing various pests. So having said that, this is all my project about. So
makes a lot of sense, and we look forward to your experiment. But remember, some of these experiments you can do at home also. Correct?
yes, sir.
So please do take up some home experiments. Don't wait for coming here or lab. At least start a few experiments which you can do at your home. All right?
Okay, sir.
You know, researchers they're impatient. You understand, Tarumai?
Yes, sir.
Researchers are supposed to be impatient with inertia. All of you should be impatient with inertia. Please remove this now and remove your presentation. Anybody wants to make any comment to him, Tarumai? Any student wants to make any comment? Anybody wants to make any comment? Any other student wants to make any comment on what Tarumai has presented? Now, this is a problem. Peer feedback, peer review is one of the most powerful means through which science has progressed around the world, not just in India. And wherever peer feedback or peer review is weak, quality of science goes down.
So I have something to ask.
Go ahead. Go ahead.
So I saw that most of the research is based on what the farmers' practices are, but I feel that a little bit of the scientific explanation behind it is not up to the mark, because we should also know what is going behind, like what is the chemical level, what is happening. So it would be better to purify or extract the product, so over here we are just directly using the citrus, the plant, but
Rishi, let me intervene. There are two aspects of it. One is affordability, accessibility, and availability. The one dimension is this: are solutions affordable, accessible? That means, are they available in local language or wherever I live? If they are only available on internet, and if I don't have smartphone, then I can't find it accessible, isn't it? So affordable, accessible, and available. So are the ingredients around me available? Do I have lemon leaves or lemon fruit around me? And if so, then I can do an experiment. So one aspect is, does it work? And if it works, can I make it work in my field? The second aspect is, which we mentioned, what is the science underlying it? Now, the issue of purifying And extracting the compound is a long drawn process. Do you know that our entire drug industry has not developed even one or two novel compound-based drug in the last 70 years? Do you understand? It's not that easy. For whatever reason, they have huge resources with them. 100 PhDs were there at one time with Dr. Reddy's lab. No, some labs are doing. Biocon is trying to do. Few others are doing, but very little, very few, rare. Ignorable. Not even one. You can't count them on one hand, or one finger for that matter, if you want to count. So surely publications will follow with your analysis and everything else. People may not benefit. So I'm not against finding out science. By all means, we should find out. By all means, we should verify if we need to. No harm. But remember, after you find out the exact compound, will you extract it from natural sources, or will you synthesize it? That will make a difference to its cost. If it is so costly, the compound becomes so costly to access that people can't even benefit from it, then what is the point of that science? Yes, curiosity is fulfilled. Please, by all means, we should do that. But did it solve the societal problem? No, it didn't. So a lot of our research is publication driven, which is, I'm not against publication. We must have top quality publication that gives respect to our science. But by itself, that's not enough. In BIIS, we would like every research to be translated. So publish well, but also provide it well to the people. So Arushi, your point is that first, why will you do the analysis? Many times we start doing separation and purification of compound without even knowing whether the practice is effective. So first task is what he's trying to do, to find out whether it is effective. Once it is found that it is effective, it's not necessary that there may be only one compound which will be there. There are 4 elements of any solution, any drug, any biochemical solution. Curative, number 1. Number 2, carrier. Number 3, suppressant of side effect. Number 4, bioenhancer. These are the 4 components. They may not always be available in one plant or one source. And that's where the science of formulation comes in. For example, piperin from pepper, black pepper, is a useful bioenhancer. So a lot of Ayurvedic drugs will have this because it increases the effect of whatever compounds are made available in that formulation. Bioavailability is increased. So please understand that formulation of a solution is our ultimate goal. Finding the ingredients and, if possible, the pure form of compound, by all means, we should do that. If it is affordable for the people, and if it is effective, and if it can be manufactured at a very low cost, by all means, we should do that. Otherwise, a crude extract, as Ayurvedic drugs often use, not bad, if it solves the problem. Today, in systems biology approach, people are coming back to the point that systems biology will require that there are multiple targets to which a molecule affects. Now, what is the side effect? Unintended target. What is the crude drug doing? It has hundreds of molecules. And they are performing all one of the 4 functions that I mentioned: curative, carrier, availability, and bioenhancement, and suppressant of side effect. So, Arushi, do you follow what I'm saying? Did you understand?
Yes, sir, I do. I will take this as a feedback.
I'm not against finding out purity. Your point is valid, that we must not leave it only at the empirical practice. Then what have we added value? Farmer was doing that anyway, isn't it? So you're right, that we should go to next step, which is to make it more effective. That I agree, because everybody may not get lemon in all the seasons. Everybody may not get the leaves of lemon everywhere. So there must be a way in which we can extract, make it storage stable, and supply it to those who don't have access to lemon in their region or in their farm. So at that point is valid. But purification, yes, may be necessary, may not be necessary, depending upon the case-to-case method. That's all I'm saying. All right, next, please.
Anjali?
Yes, sir.
Yeah.
Yes, sir. So I will start now screen sharing. Sir, before only I contacted the farmer, and for biopesticides, they were using the Pongamia seed and neem seed kernel. So, sir, what I thought is that before I did the one project of isolation and screening of bacteria that is having capacity to degrade the heavy metal. So, when yesterday I went through screening the different parasites, I got to Vorticella convallaria, which is a protozoa, and it is having a great capacity to affect like gut of the animal, and it can be act as a parasite which can remove this, means it can act as a biopesticide. So I want to add this protozoa with neem seed kernel and Pongamia seed and to prepare a solution. And I also want to see the activity and action of this protozoa, how it is acting as parasite or insect, like any big insect which are sucking or flying insect as well. So hello sir.
Do you have facilities in?
Hello.
Hello. Do you have facilities for doing it in your college?
No, sir. In my college, I don't have this facility.
So then you will have to come here, and basically you want to know, but
yes,
because
sir.
both Pongamia seed and Pongamia leaves,
neem seed kernel, sir.
Neem seed kernel, all of these are very well tested. In thousands and thousands of papers are available. So my suggestion is,
yes, sir. But in that's protozoa, sir. Yes, sir.
No, also protozoa. I mean, if you search for that, you will find it out.
Yes, yes, sir.
My only suggestion is,
yes, sir.
that if you search for the papers and if you look at whether neem has been used for protozoa control,
yes, sir.
you may find them. All right. Now, at that time,
okay,
you will see whether what
yes,
you're
sir.
going to do is novel or not. My suggestion only is there must be some novelty in whatever
yes, sir.
that we are not going to compromise with. And if we have to ask you for one more revision, two more revisions,
okay, sir.
we are going to ask you for. But when we ask you for revision, we will send you some references so that you should know why we are asking you to revise. All right.
yes, sir.
But the problem is genuine. Now, what happens
yes.
sometimes is that the method of extraction can make a difference. Supposing the papers that are there, they have used solvent-based extraction and they have not used water extract. Let us assume for a minute. Whereas if you are using water extract,
yes, sir.
and if it shows properties which are good enough, but then you have to check it shows properties for how many days, 1 day, 2 days, 5 days, 1 week, 1 month. And if it is every time you have to make it fresh, because it doesn't prove to be effective after 1 week of storage, or it develops fungus. You know, neem seed also gets fungus. It might be surprising to you that while neem is used as a fungicide, but neem also have neem seed, if you don't dry it properly, develops fungus. There are fungus which grow on neem seed, and therefore neem seed quality goes down and oil yield goes down. So you can read about it. So my suggestion is try to bring more novelty either in the process, either in the method, material, application, or business model. One of the four should be new. All right. Next, please.
Okay, sir, can I tell one more?
Go ahead.
Sir, when I discover like thermicompost, sir, here in Bangalore, there is one NGO like Kalika Kendra, and they are, sir, producing compost using a dry waste or like household or the leaf which like on the roadside. So I worked there as volunteer. They were like not using that big piles of woods are there, like sticks, woods, big one. They are not using that for composting. And that was challenging for them. So, sir, like what I thought of, like termites are there and they're having great capabilities. What I studied, get to know that they can degrade like cellulose like material also.
Correct.
And sir, also the in sericulture, when my friend is having a great farm of sericulture, he told that the sericulture many waste are there, like some mulberry leaves and many things. So they also use it, like they use that composting. They add that sericulture waste or it's mulberry leaves and all. So what my idea is, sir, that we can do like sericulture waste we can take. And sir, also like Pongamia, I've studied that, sir, we can see many leaves are there in winter, like they are here, they are on roadside, they're fallen. So what I am thinking is like we can take the sericulture all waste and Pongamia leaves, we can dry them. And sir, from that, like many ragi stalks sir can also be used, because in this south where I am, many ragi straws are seen. Ragis are very famous here as a food. So like ragi straws, sericulture waste, pongium, these all can be taken, and like they should be kept in a like container. They can be mixed with powder form, they can be dried, and that termites can be used in that. So, and for their
Yeah,
support
yeah.
on
No,
that, we can easily
but
add red soil.
termite is very
Yes, sir.
good. Not only termite, but even the termite mound soil.
Yes, sir.
Termite mound soil
Yes,
is
sir.
also very good. All right. So there are many papers
sir. Yes,
available
sir.
which have used, which have explored the use of termite for composting residues like stalk, roots, and other such things. So please go ahead. I mean, that's a good thought, and it is well known. And that group that on you can also do some experiments, and it is worthwhile, because why should they leave them?
Yes, sir.
Yeah. So there was a farmer in Kutch who
Yes, sir. I will try using that
yes,
pongemia
and
leaves
in
and
our database
sericulture
also
waste
you
and
will find
also in mass.
in our database also you will find many examples. Okay.
So, sir, can I work on this also? Like, I can bring more
Well, there
knowledge.
is nothing.
Like, I
This is a
can
well-known
see
practice,
more.
Anjali. You can do that. I don't mind, but it's
Yes,
well
sir.
known. It is well known. Only thing that you can do is maybe
Okay, okay, sir.
standardize the density, duration, diversity. These are the three things that you can standardize.
Okay, sir.
So whether mixing these twigs of different species
Okay.
helps in termite growth, or is it better that you keep all of them in separate heap and then use termite? Those kind of experiments you can do. Density, duration,
Okay, sir.
and diversity. These are the three aspects which will become very important for any biocontrol agent. Okay? You can do that, no
Okay,
problem.
sir.
Why will we prevent you from
Thank you,
doing
sir.
something useful? Never. All right, go ahead. Next one,
Yes,
please.
sir.
Prithvi?
Yes, sir. Hello, sir.
Yes. Go ahead. Go ahead.
Sir, my sharing is disabled.
No. Please, Amar.
No, it's open for, sir. It's open only.
No, sir. It's not open.
Yes.
अमर होस्ट ही कर सकता है, ऐसा लिख के आ जाता है। हर एक को परमिशन नहीं दी हुई है ना।
I opened for all the participants.
अमर मैं जब कर रहा हूँ तो only the host can share in this.
तो ऐसा करो ना, उसको चेंज कर देते हैं ना, अभी रुक जाओ। शेयरिंग ऑप्शन को चेंज करते हैं। कहाँ पर है शेयरिंग ऑप्शन? So Prithvi, please start yourself. Please don't wait for the presentation. Please start. You have limited time. Prithvi,
Yes, sir.
please
Sir.
start. We are doing that, but in the meanwhile, please start.
So
Or
yesterday,
send it to us and we will share it.
yesterday, me and my classmate Rakshit Gowda had prepared proposal on mosquito and ovicidal properties of Pithecillium dulce.
Yes.
We have done the literature survey where results showed that extracts possess bioactive compounds with significant antimicrobial activities. Methanol extracts showed highest larvicidal activity, and results suggest that the seed and leaf extracts have the potential as an eco-friendly option for mosquito vector control.
Okay. Now, what is new that you're going to do? This is already known.
We are thinking of adding Lantana camara and marigold according to their efficacy in larvicidal and ovicidal activity.
Ginny, what did who was that student Tanmoy who mentioned about database? Tarumai mentioned something in his revision which you did not notice so far. What did he say? What did he say which is relevance for Prithvi, for you? What did he say?
Tarmoy?
No, Dharma said something which Prithvi must have learned, listened to, and we said everybody can use that approach. What was that approach? Prithvi, did you listen to him? Do you remember?
Yes,
What did he say? What did he do? Tell me, if you listen to him. Huh? Forgot? Or did you not listen carefully? My dear friends, it is not even 1 hour. Only half an hour before Tarumai presented. And if we will forget so soon, then learning will be affected. What he said was that he found the compounds which were reported for a particular action. Then he searched which other plants had those compounds. Correct, Tarumai?
So it was actually me, Ayush.
Oh, sorry, I'm sorry.
Ayush.
Ayush, okay, sorry. So what Ayush did was, he found the compound which were responsible for an action. Then he found which plants had those compounds from the databases, and thereby developed his formulation. Prithvi, you should do the same thing.
okay.
Then your search for supplemental plant will become easier. And one treatment, you can keep this particular plant as alone, only one ingredient. Okay?
Okay.
But other treatments can include more than one. Okay?
Yes, sir.
All right. All right. Anything else that you want to say?
No, sir.
All right. Good luck. Next one,
Thank you.
please. You are based in Bangalore, Prithvi?
Mysore.
Mysore, okay. Because there is a group in Tata Genomic Research Center at in Bangalore, and we are doing some work on this particular problem with them on larvicidal plants. So if you, at some stage, you have extracts which have to be screened, we can get you connected with them. All right.
Yes, sir. Thank you, sir.
Next one.
Saurabh?
Yes, sir. Yesterday I revised the proposal and I came up with an idea for making a portable herbal decoction machine. So currently, herbal decoction machine, sir.
Okay, very nice. It is a fuel, wood powered, fuel powered, or solar powered, or electricity powered?
Electricity powered. We will use a 12-volt 2 ampere adapter to power the motor for crushing the herbs, as well as it will consist of heater at the bottom for heating the mixture. So currently the power outage is currently, we will step up the power, and it can be used by office workers. So it is being a portable, and I searched on the Google. I was not able to find a particular product based on herbal decoction machines. Two or three literatures I found patents also, but the main issue there was they only had heating properties, or somewhere only grinders. So we are, I am thinking about first, like the process, we know that first we have to crush it. Then after crushing, we will pour into the vessel. And then after that, it will measure what is the weight of the herbs. And as per that, we will pour the water. And when the desired amount of water is achieved in the container, it will trigger a signal like a buzzer or a LED, which will indicate that this much amount of water is required, sufficient. Now we will stop, and later we will just switch on, and the heating will start, up to which the desired amount or specified amount of water is reduced. Like I studied the Ministry of Ayush website, it was about 1/4 of the original mixture we have to get for concentration.
This will vary from case to case.
Yes.
But are you trying to talk about, you're not talking about distillation apparatus, you're talking about boiler basically, is that right?
Yes, sir. A grinder, a boiler, in a single device, a compact device, like just a shape of a bottle.
Okay. So
because
boilers are available. What is new in it? I can't follow.
So the main problem is that it is a cumbersome process. So currently, no technology as such that in market, no product is available.
No, what are the features of this product? What are the features of this product?
So currently,
it will
currently
crush.
I'm not thinking. Yes, sir.
Will it crush, boil?
Yes, sir.
And
First it will
complicate all
crush.
the three things.
Then,
Is that what
yes,
you're
sir.
saying?
First it will crush, then it will boil. Then, as per the desired amount of herbs which you have weight of the herb, it will automatically add the water if you add the solenoid, and then it will filtrate through with a simple filter paper attached at the wall at the bottom.
No, with filter paper. I mean, you can have cloth piece or something.
Yes, cloth filter. Yes.
Okay.
So it is a
right.
portable. We can use it in the we can carry it to the office, or it can be used at tea stalls. So it will also promote traditional knowledge. It will bridge the existing gap.
Okay, go ahead. Go ahead. But look at the grinder, boiler. I mean, one of our innovator, Dharamveer, also has developed. He has added a distillation also. So there is a grinder, there's a boiler, and he also has an extractor, which is distillation, all in one machine. But that's, of course, is bigger scale. If you are trying to do a small scale machine, that's good.
So extractor, I also I would like to add that I also saw the part of the extractor. So the main issue in the extractor is the desired amount of force which is required. So that can't be used in a portable, because if we add that mechanism, it will become bulky.
All right. So go ahead. Go ahead. Go ahead.
Okay, thank you.
And then, but you should have a user in mind. I mean, who will like to use it? That you should have in mind. Is it for making a fresh soup? Is it for making extract in the tribal communities? And then you make it battery powered, so battery can be connected. It can have mains, or it can be used as solar also. If it is a low wattage battery, 12-volt battery. If it works on 12 volt, that's good enough. That's portable.
Yes, I am thinking of installing a small lithium power at the bottom, so that we can use it anywhere, and we can charge at home or something like that.
Is it a pressure bound, or is it open pan?
Sorry, sir, I didn't get.
Is it a pressurized boiling, or it is open boiling?
So open boiling, so not pressurized.
Okay. So you can think about how to. There are some, for example, autoclave function has a pressure-based heating, but go ahead and do that. I think we should go step by step. All right.
Okay, so I will check also about pressurized boiling, and if we can add, I will look into how can I integrate that feature.
All right, go ahead. Next, please. Next, please.
Anyone else revised your proposals? Can present.
Nobody else is there?
I guess so.
Sir,
yes.
So I would like to ask something that I have done a home experiment at home, like the kitchen gray water. I filtered it with a carbon filter, and by pouring it into ceramic powder, and then added neem extract and observed the growth of plants.
Mm-hmm.
And
kitchen water, you don't have to filter, because kitchen water will have nutrients mostly, isn't it? Unless you are washing using detergent, then it is a different matter.
Yes, sir. Yes, sir. The soap material is there in the.
Yes, so if it is soap material is there, then I'm not sure how effective the charcoal filtering is, but if it is doing well, then it's okay.
So if you permit, I would like to share the screen so I can share the photos of how.
Okay, sure. Share it. Permit kar do bhai.
It's open only, sir. He can do.
Saurabh?
So no, it is showing host disabled participant.
Okay, so please
Just a
enable
minute.
everybody. Enable everybody. अमर वहाँ जाओ ना जहाँ पर enable का बटन होता है. मुझे क्यों नहीं दिख रहा है वो आज? Advanced sharing option. Okay, let me do that.
So yes,
All
is it
participants.
visible, sir?
Yes.
Now it should be
Yes.
open. It's
Yes,
all,
yes, yes.
all participants can share it now. Yes.
So first, I just took a small micron mesh to filter out the food particles, and then use for activated carbon powder to trap the molecules, and pour a ceramic bunch of them in a single container. And then I used some neem extract in the water. This was the CAD model which I designed. I have a 3D printer at home. I printed the outer body casings and all. And this is the model, which it is a portable, very compact, small model. This was the water samples. This is the gray water. This is after treating it with carbon filter and ceramic. And after that, we added neem to it for disinfection. And this was the results. So, as you can see, neem showed a significant amount of growth than gray, the pots with gray water and the filtered water.
Very good experiment. Very nice, simple, straightforward, and visible result. Very good. Very good.
This water can be
when
used
did you
for
do that? When did you do that?
At home, sir.
In what period?
So in these 15 days of.
Very nice. Very nice. Has anybody else done any home experiment? This is very good. Very good.
Sir, I have started, sir.
Tell what have you started? Thank you. Thank you. What was the name presented just now? Amulya?
Saurabh.
Good effort. Very visible results. Now the question is how costly the filter is. Can you make it cheaper?
So the whole assembly which I have designed is of 900 rupees.
900. And how much time it takes to filter 1 liter of wastewater?
So currently I took a filter which was wasted in home. So that was used a filter, and it filters about 1 liter for about 20 minutes.
20 minutes. That is quite long.
It is a waste filter, but we can.
No, I understand. My suggestion is we will give you more resources if you want to come to our lab. Our incubator will incubate this solution. Okay.
Yes.
Chetan? We will incubate this solution. You make a product and we will help you to launch it, because many people need a ready-made product. Now, only thing is that you should have a cartridge-based system.
Yes,
so if you want to change you should work out at what frequency the charcoal has to be changed, at what frequency the neem seed extract has to be changed, correct?
Yes, sir. Filtration capacity, no?
Filtration capacity and filtration saturation.
Okay, sir.
Saturation also has to be more. So you will have to do some experiments. Now we are getting Professor Shekhar Mande, so we will all get to hear him. Hello. हाँ, तो मैं आपको कर दूं स्पीकर फोन पर?
अभी नहीं, 12 बजे के करीब आने से चलेगा?
12 बजे चलेगा, कोई दिक्कत नहीं. We'll wait for you. 12 बजे आ जाइए. मैंने आपको लिंक भी भेज दिया, आपको जूम पे आना है या फोन पे, जैसे आपको कन्वीनियंट लगे. ठीक है ना? Thank you so much. Thank you. So Dr. Shekhar Mande will join us at 12 o'clock, which is fine, not a problem. He, as I told you, in the field. So carry on. So what I'm saying, Saurabh, was it Saurabh?
Yes, sir. Yes, sir.
Saurabh, my suggestion is that do you want to take it up as an entrepreneur, as a startup?
Yes, sir. Sure, sir. I would like to innovate products. So,
all right, then we will accept you in our incubator. Okay. And we will support you to develop a market-ready product. Please submit a detailed proposal. Okay.
Yes, sir. So I had a doubt, and I would like
Go ahead. Go ahead.
So जो neem extract है, उसका एक filter में conversion. So how can we make that? Like I have seen.
देखो ऐसा है, neem extract. One way is to have a layer of neem extract or neem powder, seed powder. Second is to, within the charcoal, The second way will be to mix it with the charcoal. Okay.
Okay.
Third will be that you take some other material which is saturated with neem extract or neem oil. So there are different approaches, because if you take, let us say, a porous material, say chalk material or some such material which is inert, but which can be saturated with which has filtration, which has capillaries, and which is saturated with neem extract. That material can be added in between charcoal or after charcoal, as the case may be. The question we have to standardize is how much time would each of the column would need for filtering the water effectively. And that is where the research is, that is where the product development will require, and we will help you to do that. So if you have to take 10 filters and spoil them and do the monitoring data, all that is possible, not a problem. But these are questions which you are asking are the questions that you will have to answer by doing research experiments. Isn't it?
Yes, sir. Sure. Sir.
Let us do that, and we will get you to get in touch with Professor Pradeep at IIT Madras. He is one of the most renowned scientists in our country on water filtration. And he's a dear friend. I'll request him to mentor.
Yes, sir. Thank you, sir.
All right. Yeah. So you will get mentoring from Dr. Pradeep and his team. Maybe one of his doctoral student or postdoctoral student will also help you. Once you revise your project along with his photographs that you have done, send it to us, and we will send it to him and put you in direct touch also, so that if they want to give you feedback directly, they can give you, or if they want to tell us, we'll do that. And if need arises, I can send you to IIT Madras.
Yes, sir.
All right, you can work in his lab also in summer vacation. When are your exams ending?
So I think May 1st week.
May 1st week. We will organize that. All right.
Sure, sir. Yes, sir. Thank you, sir.
Good luck. Good luck. This offer is available to each one of you. Anybody who develops a product and wants to take it to the market, and in summer you want to do some test some waters or develop some product, so that in your next phase of life, whether as a postgraduate student or as an entrepreneur, you want to do more, we will encourage you, we will support you, whatever is your direction. So long as the product meets a real need of our society, and it is affordable, and it can be, and you are willing to try and develop it. All right. Anybody else who wants to present? Now, Amar, we can allow, with reluctance though, we have half an hour. Okay. So if anybody else wants to share their ideas, we will allow them with the reluctance, but okay. And Professor Shekhar will join at 12. Amar?
Professor, yes.
Yeah. Let us see if anybody else has. Can anyone of you?
Anyone else who wants to present, who haven't presented yesterday, want to do?
But the idea must be backed up by some homework. I don't want raw ideas just now. Some homework. Raw ideas we discussed yesterday. Now we want ideas which have been processed.
Anyone else?
Anybody, please go ahead.
So I have done a synopsis, but I feel it can be better. So
okay,
that is why I won't be presenting today.
All right. You can briefly present now, or you want to do it only later?
So can I just talk about it? I have done the research, but I just want to talk about it.
Go ahead.
So I would be I'm proposing that we use the shoot of Boehavia diffusa to treat the inflammation caused by arthritis.
Yes, continue.
Go ahead, please. Go ahead.
So basically, sir, there are about 210 million people in India who have arthritis. And according to some research done, 7% of the people are reluctant to go to doctors. And a lot of these drugs, inflammation treating drugs, are very expensive and cannot be afforded by a lot of people, because arthritis is both prevalent in rural as well as urban populations. because of the increasing contamination in the water, and due to since there's mineral deposition in the water caused by industrial pollution, the arthritis cases have been increasing in India. Hence, I want to investigate the one of our farmers has proposed that he uses the shoot of Beauveria diffusa to treat inflammation and arthritis as a one-time remedy. Hence, I want to investigate this particular grassroots practice so that we can make this more reachable for people who are facing the issue of arthritis.
Which plant did you say? Diffuser. What the first one you said?
Borhavia.
Borhavia. Okay. Yeah. Now, have you not found any solution? Any literature?
So this is basically used in a lot of diseases. It is a very popular plant in Ayurveda.
Yes.
It has a lot of properties which treats many. There were a lot of diseases. I have to pull up the list of it. But arthritis is not explored yet. And the thing is that different parts of the plant is used for different diseases. So over here, I'm proposing the shoot.
Let me share this paper from a very ordinary college from botany department, Harpreet Kaur. And she has mentioned arthritis also for this, I will share with you some more papers. My suggestion is that your life is precious, isn't it? You are only of your kind. Is there another clone of yours? Wait a minute, I'm getting a call from Dr. Shekhar. Yes, sir. Yes, Dr. Shekhar. हेलो, हाँ हो सकती है ना, तो अभी आप फोन से करेंगे या मैं जूम पे करें, जैसे आपको कन्वीनियंट। अच्छा ठीक है तो मैं एक मिनट उनको let me just put it near the. So friends, we have Dr. Shekhar Mande, Director General CSIR, a very eminent scientist, very valued, recognized and respected leader of science, who has galvanized the entire CSIR community to bring tremendous amount of research and outputs for social impact and for industrial impact. So I will not take any more time. Shekhar, you're on and everybody is hearing you.
Oh, thank you so much. And I'm so pleased to be amongst all of you today. Unfortunately, I'm not there in person, nor I'm there on the web, on Zoom or something. I'm on the roads, but still it's a pleasure to join with all of you today, this morning. I'm told that a large number of entries were received, and out of which about 30 or 35 entries have been actually shortlisted to be brought here. And we are trying to make provision for funding the most exciting ones of these for some research for future. I don't have to say too much about the role of biotechnology for future. What I would like to know is that it's recognized all over the world now that biotechnology literally pervades across all disciplines of human endeavor. So, for example, if you want to improve the quality of crops in agriculture, or you want to see what kind of food we are eating and whether that the food is good for our health in nutrition, biotechnology of health itself is a completely different area. That for protection of environment and general biosphere. So think of any area and biotechnology appears to actually pervade through all those areas. And our understanding, unfortunately, of biology, life sciences, or our biosphere itself are not really up to mark when we compare ourselves with, let us say, other disciplines like that. So the other disciplines have made great progress in the last 200 years in improving our understanding of those disciplines. But the fact that biology was actually a late starter in the history of science, and another fact that biology itself is so extraordinarily complex, has resulted in not actually having optimal understanding of everything around us. But yet, the applications of what we have done in our research and all, for the benefit of mankind or for the benefit of the planet, are truly amazing. in this exciting development. All of you, I hope, would keep up your interest in biotechnology for the coming years, and all of you, I hope, will contribute in the coming years in the improvement of our understanding and thereby improvement in general well-being of everyone. I've not heard what kind of projects you are doing, but I will definitely catch up with the to see what kind of different projects that you have proposed to do. And in taking those projects to some sort of proof of concept, if there is any support required from our labs or support required from mentorship of our scientists, we'll be delighted to help. CSIR, many of you would have heard, is one of the largest civilian R&D organization in the country. CSIR spans many different disciplines, starting from mining, chemicals, aerospace, and so on and so forth. And because we are actually in almost all the disciplines, if any discipline benefits by all of these expertises together, is of course biotechnology. And that's because biotechnology itself is a very interdisciplinary science. So any mentorship or any help required from CSIR labs, as I said, we'll be delighted to extend that help to all of you. Because we are actually present in all the different disciplines, I can also share with you some of the excitement that we are going through. As I speak, I have been in Palampur for the last couple of days, Palampur in Himachal Pradesh, where we have our lab, Institute of Himalayan Bioresource Technology, IHBT. It has been doing some extraordinarily good work for the people of the region and in agriculture and other biosphere-related activities in general, in documenting Himalayan biodiversity and so on and so forth. What I witnessed in the last two days has been something really truly extraordinary, and that is the IHBT Palampur decided to cultivate tulips in India. As all of you know, tulips have a great potential both in terms of floriculture and both in terms of promoting tourism. And in India now, the reported from the Netherlands import of the order of about 5 to 7 lakhs of tulip bulbs every year from the Netherlands. But IHBT decided to change that course of history, and they have started cultivating bulbs in areas of the country, such as Lawrence, Spiti, and Shaka. And the first result of that is what actually I witnessed in the last 2 days in Palampur. The tulip garden looks just simply amazing. Similarly, as we speak, I also have to bring to your attention that the country's first two-seater trainer aircraft is undergoing its regulatory process. We are almost at a turn of an event where the last of the flights for regulatory requirements completed its flights yesterday, and we are now ready to go to the regulator to get the final certification. This is an indigenously developed aircraft, and the aircraft can be used for training our pilots who can eventually get commercial pilot licenses. Now, the reason that I'm telling you these two stories is that the breadth of work that happens in CSIR is enormous. Much of the focus of CSIR's research has always been to generate Atmanirbharta in all our endeavors. There are two examples I told you about the tulips and the aircraft. But the true Atmanirbharta would come in future, not in adopting the well-known technologies for our country's development, but rather develop the technologies ourselves for the future. Which technologies the rest of the world will also use from us. And that actually would be the true dream of Atmanirbharta coming through. We must actually now operate at a global innovation system. We must be leaders of global innovation in our country. And to that future, your generation has a lot to contribute. And we very fondly watch you in the coming 20, 30 years as you make progress in contributing to the global innovation ecosystem. The fact that you are here today as the chosen few from the competition that was held nationwide bears testimony to the fact that all of you hold promise to the future of our country. And as I said, we will keenly watch your progress in the coming years. You being at a BIS meeting and getting these awards is only the starting step. And there is a lot more ground to cover. And I wish you, all of you, the very best into that future and into that contributions that would come from people like all of you. Thank you all so very much, and all the very best for your future endeavors.
Thank you
Thank you
so much, Shekhar. I will just briefly mention that some of them are doing very practical and very realistic projects. Some of them have taken up the traditional knowledge of their own elders, their own grandparents. Some of them have taken up in very interesting innovations from our database, such as use of neem, such as lemon leaves for pest control and as growth promoter. Some of have taken up, they have taken in devices. So one of them is trying to filter with charcoal and neem seed extract the water, wastewater of the kitchen. And then he did a home experiment to show us that when you use only water as the kitchen waste, kitchen water, or you use it with normal filter, or you add the neem extract to that water, the growth of the plants is so discernible, different, that it makes a lot of sense. So there are doing some very good projects which are touching the very real problem all of us face in our own homes or our farms, and I will keep you posted with that. Thank you so much to be with us. And all these students are tremendously inspired and geared. Your offer of mentoring by the CSIR scientists is very, very welcome. This was one question they asked, and I'm sure you will connect them to some of them. Thank you so much. Thank you so much.
Thank you so much.
We're grateful. Thank you. Thank you. Thank you. So friends, you were just listening. I hope you could listen to him. Was it all right?
Yes, Professor.
Right. Thank you. Thank you. So you could see that Director General CSIR was addressing you and assuring you, and also mentioned that he will be watching you as to what you have done and how you are going to make a difference to this country, and how keen he was. And I showed you the picture of the small video of the tulip also, which IHBT has developed. I couldn't show you the Nels flight of the airplane on the sea, which was also achieved yesterday. But that only showed that the wide range. I was at CMARI a few days back at Durgapur. I missed Tharma there. And I can assure you that CSIR scientists will be very happy to mentor you, apart from these scholars from IIT. I'll be meeting Amresh on Saturday, 12th in Bengaluru. And I'll be meeting, I'm Bangalore director also on 13th. And both of them have assured me that they're support. So if any one of where are you based, Saurabh, the one who was developing a water filter?
So I'm currently living in Sangli.
Sangli,
Maharashtra.
Sangli, Maharashtra. But where is your college? There only?
So Karad.
Karad. Okay. So if you need help from NCL at any stage in Pune, we'll organize that. I mean, I already offered about IIT Madras, but we can also arrange your visit to NCL for getting some advice. You know, that would not be a problem. But
yes.
Okay, Amar, what is the next program now? Is there anything else? Okay, vote of thanks. So before you thank everybody, let me just take one more minute to say that we are so excited that all of you have been able to walk with us for 3 weeks. You are now ambassador of Honeybee Network, Shashti, Jan, and I have all over the country. And Shashti has invested in databases. Shashti has invested in the lab. Shashti has invested in creating a network of mentors. And I hope that we'll be able to support your future aspirations as you go along, particularly of those who are going to work hard in translating their ideas into practice. We are very concerned and we will be very happy to support every student. As I've said, even if you don't qualify for the World Like Award, we will still like that you are able to come to our lab, work with us, stay here, develop your solutions. And obviously, in the process, when you come here, your proposal may go through some revision because our team will like to make it more efficient, more effective. And we are hoping that you will all be with us as and when time permits. Please aspire for larger transformative impacts. Don't think small and don't think too short. Think in long term, think large. And I'm sure that you will be able to say, Chetan, are you around? Do you want to say anything? Chetan, are you around? No, I think he's left. So friends, welcome once again to this new journey, new phase of your life when you will be a volunteer of Honeybee Network, you will be an ambassador of Srishti and BIIS program. BIIS and DBT, BIIS is a subsidiary organization of DBT, Department of Biotechnology. BIIS is investing in this program because they want to create a pipeline of future translators, the future scholars, young scholars who will translate science into technology practice. And as I already explained, I will not repeat, many of you have many options of, you should keep looking at BIIS site for other fellowships that are available, other opportunities that are available. And anywhere you need a channel to pursue your applications, we'll be very happy to mediate and provide you all the support. So good luck to you. God bless you all and do well in your life. Amar.
I invite Dr. Premlata, Srishti GAITI coordinator from Srishti Lab to deliver the vote of thanks.
Go ahead. I'm not getting any voice from her, Amar. Either
Doctor,
she comes to your...
Premlata.
Either if she's here, then call her to your own computer. If she is at home, then we are...
She's at home.
Then I think you should give the vote of thanks because I can't hear her.
I take this opportunity to thank Professor Anil Gupta.
Don't thank me, thank all the speakers who have come. We don't thank our internal people, but go ahead.
Thank all of our guest speakers from various institutes from the India and all the students who have participated enthusiastically in this 21 days workshop. We thank BIIS, Dr. Manish Diwan and Dr. Shilpi Kochar for supporting us through this program to identify and promote the young entrepreneurs and their ideas to develop them into future startups, to develop into startups. I thank all who have supported us to conduct this 21 days workshop very smoothly and be productively. I thank all of you. Thank you. Thank you so much for joining us.
So good luck to you all. Bye-bye. God bless you all and do well in your life. And we are looking forward to receive your detailed proposals and hope to get 10 of you to the next level. Thank you so much. All the best.
Thank you, Professor.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.